Welcome to the Genetics Podcast. As you may have heard, we're running a new mini-series called Precision Pioneers, which is focused on the people's, companies, and other organizations that are at the forefront of precision medicine. Our second episode in the mini-series is with Dr. Nihar Bhakta, who's the Chief Medical Officer of Aristea Therapeutics, which is an immunology-focused drug discovery company. He spent much of his career at the forefront of precision medicine. I'm really excited to be here with him today. So thank you, Dr. Bhakta, for taking the time.
No, thank you very much, Patrick, for having me on. I really appreciate it.
Of course. So I'm wondering if we could start with your first exposure to personalized medicine. You've been working at the forefront, as I mentioned, for your entire career. So maybe we can just go back to the very beginning, to the late 90s, early 2000s, when you were in medical school and residency, and talk about your first exposure to personalized medicine and maybe a little bit of how it's changed over the years.
Yeah, absolutely. Absolutely. So I started my medical career at the Cleveland Clinic during my residency. And I think that was probably the first area where I started really being exposed to rare diseases, personalized medicine. I did my residency in pediatrics, and there are so many rare diseases that only occur in pediatric patients. And I was, at a training center where we saw, a lot of different diseases, but we also had a lot of people who were referred in because they did have very rare diseases, certain mitochondrial disorders. Obviously, we had tons of cardiac specialists at the Cleveland Clinic, so we saw a lot of rare congenital heart disease patients. Many of those were, obviously genetic in basis, but some were, had other etiologies as well. So it was a great place to train. And then subsequent to that, I decided I did want to do specialty training. So I decided to do a fellowship, and I did my fellowship at UCLA, another great center where there was a really good referral base, and I learned a lot there from I had a ton of really good mentors, and we really pediatric nephrology is a very interesting space in that there are a lot of diseases that truly do have a genetic basis. And it's incredible how some of those diseases, even the care of those within, the time I've been practicing and over the last 20 years, even that has changed. One really good example of that is a disease that we actually got very expert in, which is a very rare disease called primary hyperoxaluria type 1. So this is a very interesting disease in that it's actually a disease of the liver, but as nephrologists, we cared for these patients. Basically, these patients have a mutation in a specific liver enzyme, AGT, alanine glyoxylate aminotransferase. And in essence, what happens is that this deficiency results in the accumulation of glyoxylate and too much production of oxalate and glycolate. So, what's important here is that oxalate is actually in the form of a calcium salt that's very insoluble and gets excreted by the kidneys. So ultimately what happens is that these patients actually have deposition of this oxalate in the form of stones in their kidneys, and then they subsequently go on to renal failure. And so we manage these patients as renal failure patients, even though their primary defect is in the liver. And so when they come for transplantation, they actually need to have both their liver and their kidney transplanted. And we saw, I want to say, well, during the time I was there, I think we did at least 6 cases like that during my training. I had one weekend where I had 2 patients on the same weekend get both livers and kidneys. It was a very interesting case just because these were very young patients. And so they actually had the ability there was one donor and they split the liver and they gave a piece of each liver to each pediatric patient. And then they gave them each one kidney because when you get a kidney transplant, you only get one kidney. And that happened on the same weekend. And, just the acute management of these patients. And it's incredible because, the team there had really developed this protocol, which they subsequently actually wrote up in a very nice fashion because they wanted to really try to standardize the care of a disease that's, very rare. And they wanted to be very consistent with how they did it. But the problem, as we know, is it's hard to run clinical trials. How are you going to run a clinical trial where you're comparing how you manage these patients versus maybe another methodology for managing these patients? And I think that was one of the first times where I really started thinking about how challenging it is in rare diseases to conduct appropriate clinical trials. And so much of it is really based on experience to start with, but then you have to start evolving that, right? And I think that was probably the best lesson that I learned while I was there is that you really do need to keep pushing and trying to understand what is the best care for these patients. And that's really what drove me to want to get into doing further research, because you realize that there's a way to truly improve care for these patients. And some of it is iterative over time early on, but ultimately it does need to move into clinical trials.
Yeah, well, I'd love to go more into the clinical trials piece and what has changed for the better and what still needs to be innovated on. But as you're telling that story, it struck me that what you described with the double transplant is a form of treatment. Of course, you're replacing the liver with a healthy liver that doesn't have the genetic mutation. You're replacing the kidney with a healthy kidney, and I assume those children you'll go on to be relatively healthy. But it's not often what we think about when we think about a therapy. You'd think about, can there be a small molecule or something that we can treat the liver? But the way you describe it, it is the best, probably the best treatment available at the time.
Well, it was the best treatment, and now it's out, and that's what I meant about how things have changed so much. I mean, now there are actually RNA-based therapies that they're trying for these patients. There's a couple of different molecules that are in development right now for these patients. I think one is even in phase 3 development, lumirasan, and it's incredible because this is something that the patients, if you can identify them early enough and treat them early enough, you can actually keep them from getting the kidney failure and then keep them from needing a transplant, which is, that would be awesome. I mean, it would be incredible if people didn't need kidney transplants, right? If you could, take even this small population. And so I think it's amazing the strides that have been made in the last 20 years, just in terms of how people have really tried to focus in on some of these genetic defects and really tried to come up with novel and innovative ways of treating them.
Yeah, absolutely. And I'd like to come back to, I think we're going to spend a little bit of time talking about your early career, but I'd like to come back to what you all are doing at Aristea, because I think there's a couple of elements here. And one of them is a deep understanding of the biology, which you absolutely, I think you all have focused on and have. But that's a key ingredient. Not only do you need the technology like RNA interference or the right molecule, but you also really need to understand the fundamental level what's going on. But before we get there, I wonder if you could just talk about your transition from practicing medicine into developing new medicine. So you mentioned you did your residency at the Cleveland Clinic. And I think after that you moved to Roche. How did that come about, and what did you work on there?
Yeah, so I moved to Roche after I was at UCLA, and my time at UCLA, as I said, I was very fortunate. I had some great mentors. Robert Ettinger really tried to help me along and help me understand, how to do clinical trials. And one of the things that I was exposed to, which I was very fortunate in there, was actually a steroid avoidance clinical trial. It was actually at the time one of the largest clinical trials in pediatric patients, looking at the use of corticosteroids as part of their transplant protocol versus no corticosteroids as part of their protocol. It was a randomized trial. And that got me really interested in this entire process of running clinical trials, of how you do that, of conducting clinical research. And at UCLA, we had a really nice program where you actually went through this, learning basically how to write a grant. So we did part of that. I actually ended up writing a grant and getting one accepted during my third year while I was there. But at the end of that time, I realized, for myself at least, that I probably didn't want to spend the rest of my career writing grants. That just wasn't what I was passionate about. I loved conducting clinical trials. Obviously, the best feeling that I've had in my career is when you get a readout from a clinical trial, whether it's positive or negative, because there's that level of, I helped to design an experiment. We have patients who are so, we were so blessed to have these patients who were willing to participate in this experiment to try to advance the disease. And, now we get to see the result of that. Whether it's positive or negative, we can answer a question, right? And try to help patients. So that's been a really good feeling, and I've had it multiple times. And I had it while I was at UCLA, and I wanted to kind of continue doing that, probably on a little bit more frequent basis than I could in an academic role. And that was one of the reasons why I chose to transition to a career in pharma. So yeah, I moved to Roche, and then subsequent to my time at Roche, I moved to Bristol Myers Squibb, and that was an interesting transition, because at Roche I worked in their nephrology group, but then subsequent I worked in cardiovascular and cardiometabolics, and it's a very different field. So I worked on a molecule, clopidogrel, Plavix, which at the time was one of the only therapies that was approved for patients who had heart attacks and had a stent. It's an antiplatelet agent that helps to thin your blood. it was very interesting because the clinical trials that the cardiologists run are enormous. They have tens of thousands of patients in their clinical trials. I was used to running clinical trials with 40 patients, 50 patients, 60 patients.
Right.
The largest ones were maybe 100, 120 patients. So it was night and day. And I got this statistical training, which, took me from a level of almost nothing to where I was kind of dangerous, where the statisticians would look at me and say, please step away from that p-value, Nihar. You're don't point it at me.
We don't want to hear anything about that. Yeah.
Yeah, exactly. So, but you learn this really, this different and very rigorous way of looking at clinical trial data, which for me was awesome. I mean, that was one of the best, again, if you have a job like mine, and I'm very fortunate, I'm a scientist, so I get to learn all the time. And I had a job there where I was learning so much about a different disease space. I learned a lot from the cardiologists who I was fortunate to work with. Many of the cardiologists were very open and willing to work with someone who wasn't a cardiologist, and try to, teach me a little bit. And then, I had obviously great colleagues who really helped me learn a lot as well. And I would say that was, for me, a really good experience, because what ended up happening was that I got put in charge of, something that at the time was at the very forefront of what was going on, which was pharmacogenomics with clopidogrel. That was one of the areas where I had a little bit of experience actually during my time at UCLA. At UCLA, one of the reasons why I was interested in pharmacogenomics was because the kidney transplant medicines that we use, we used at the time, and we have a few additional medicines, but it's pretty much the same medicines we've been using, predominantly calcineurin inhibitors. It was very interesting. The classic one was cyclosporin, and another one is tacrolimus. And one of the challenges that you have is that you want to have those immunosuppression medications at the right level. If they're too high, you have vasoconstriction, so there's not enough blood going to that new kidney that's been transplanted. And if it's not enough, obviously you can have a rejection episode. And so you want to get that tweaked early on, but patients respond to these medicines very differently. And so some will get their first dose of medicine and have a really high level, and other ones will have a really low level. And the thing that occurred to me was, we do so much blood work on these patients beforehand because we need to cross-match their kidney. We have access to their genetic information. We might actually be able to determine who might be a really high metabolizer or a low metabolizer of some of these medications. And so I was interested in that. I'd done a little bit of work on that while I was at UCLA. And that little bit of work got me put in charge of this large program while I was at Bristol Myers Squibb. And, it was a fascinating time there because it was actually data that a competitor was putting out about our molecule. So they're using our molecule, they're using clopidogrel as the comparator to their molecule. And they did a pharmacogenetic analysis that suggested that, some patients didn't have a great response to the drug. Now, that obviously is always a challenge, and I think that especially in an area like cardiovascular disease, people have acute coronary syndrome. If you don't respond well to a platelet thinning drug, you can die, right? So and there's so many heart attacks in the country that it is an issue.
Right.
And so the company, it was Bristol Myers Squibb and Sanofi Aventis. They both had the molecule. So it was two companies were working with the FDA trying to figure out a plan to better understand what was going on here, because there was some data that had come out at an American Heart Association Congress. And I think that the way our team chose to approach it, which was the right way to do it, was, usually you want to just go back to the source data. You want to go back and say, okay, well, what do we know about our molecule?
Right.
We studied our molecule against a placebo control. We actually were fortunate in that the registrational trials, we actually still had we had pharmacogenetic consents from that initial trial those initial trials that were conducted. So we were able to work collaboratively with the investigators at Hamilton, and who had initially run the some of the registrational trials. They conducted pharmacogenetic analyses from the patients who had already provided consent, and we had a publication that came out that suggested that while there were some, that suggested that in their clinical data set, they didn't see the same type of pharmacogenetic challenges that were seen in that other clinical trial. Now, who's to know what was right or not? But you have to go back to what your own data is. So
Right.
that I thought was really interesting. That analysis, and that was the first piece of work that we did. That ended up being published in the New England Journal of Medicine, and it really shined a light on a couple of things. One piece of data obviously doesn't always show you if something is or isn't a problem. And even, if there's multiple sources of data, I think that's how we all should approach things, right? You can, no matter how large the trial is, you still need to, put on that skeptic's hat and say, okay, well, what do we really know? And are patients potentially being harmed or not harmed? Is there a challenge? And I think that, we, at the time, we took an appropriate approach there to try to understand the data. We obviously worked worked closely with the FDA around how the labeling would work for the molecule and things like that. But that was, that was a really good experience from the perspective of this was something that here was a molecule that was already on the market. There were lots of patients who were getting it. And then how do you subsequently move from there? The thing I took away from that entire experience was actually talking with the cardiologists. Many of them said, if I'm required to do a pharmacogenetic test before someone gets a dose of this drug, there will be harm to patients because there weren't any other molecules available that were part of their standard protocols.
Interesting.
And you can't wait, for someone's pharmacogenetics to come back before you start dosing them and treating them, right? So
Right.
It would obviously be something that would happen after the fact. So they would already go in, they'd have their heart attack, they'd have their stent placed. But at the time you place your stent, you actually want some of those drugs on board. So, part of the protocol is you get your aspirin, and now there are other molecules that are part of the protocol that people start getting. But at the time, it was you get your aspirin and you get your Plavix, and you go and get your stent placed so that you have. Right. And then they have other medicines that are also part of that algorithm that they used that would help them to not have that stent clot, as well as help obviously open up the clot that was already there that was causing the challenges.
How is the pharmacogenomic component to clopidogrel, Plavix, and others today? Is it because in some ways things haven't really changed? It's not that all of us are walking around with our cytochrome P450 gene status. So Are we in more or less the same position, or is it that what you just mentioned, that different drugs are prescribed kind of at the moment, and that's only now prescribed with a genetic test?
Yeah. So, to be perfectly honest, that is one of the things that I haven't kept up with. So, obviously you have to move on from your therapeutic areas. I do know that most of the, I have friends who are cardiologists. Many of them use some of the newer agents that don't seem to have the same type of pharmacogenetic challenges for a couple of reasons. Those other new agents, and they've shown in large clinical trials against clopidogrel that they, showed statistically significant improvements in death, in cardiovascular outcomes. So the cardiologists, again, are very data driven, and they've just moved to those molecules. And those have become their new standard of care. Again, it's been a period, that was in 2008, so over a period of 10 years, there've been changes to the standard of care, and people have just gotten better at managing these things. People,
Yep.
I think if someone has a heart attack, you don't think it's almost like having, a basketball player blowing out an ACL now. It's not the end of their career. It's not the end of your life. You kind of move on.
I'm also really fascinated by the rigor of clinical trials, and I try to explain and evangelize to other people what an amazing piece of science they often are, because in a lot of areas of science, there's actually issues of reproducibility and rigor, and you can't really tell from reading the paper what had gone on. But in a clinical trial, the level of rigor is just incredible, and it's because it has to be. I'm curious what you have taken away from the whole COVID vaccine situation and the incredible phase 1, 2, and 3 trials run in the space of 12 months, and often on technology platforms that had never been used before. What did you take away from all that?
Well, I took away a couple of things. I think the first is that, when you have appropriate resources and dedication and teams that are really willing to work around the clock, you can accomplish so much. And I'm just so impressed by the teams at all of the different companies, at J&J, at Pfizer, at Moderna. I mean, these people, I know how much work they probably had to put in. They were probably working around the clock, 80, 100-hour weeks, if not more, to get the studies up and running. And this was everyone at their companies, people to manufacture the vaccine, people to get it shipped, people to, all of these people who, no one thinks about when you think about data coming out from a clinical trial. You don't think about all of these people who have to coordinate and work together. You might only think about the CEO who gets up there and starts talking about, the trial results and that they have 90% efficacy or that no one died in their studies. But, it takes thousands of people to conduct some of these trials. And given the breadth and the size of them, I'm in awe of the fact that they were really able to deploy that. But more importantly, I do think that they conducted very good experiments, and they're now being borne out in a real world setting. I think that, I fully would expect that what Pfizer's been able to show in Israel with their real world data is going to be reproducible with the Moderna vaccine, is going to be likely reproducible with the J&J vaccine. I think that, these were well-designed, well-run trials that will likely be just as effective in the real world. Potentially, and you hate to say it this way, potentially even more effective, given the fact that now there are certain pockets of the world that do seem to be developing some bit of partial immunity, right? I mean, if you had enough people, and then you get enough of those, people who live in that area who've gotten it, and then a bunch of other people who are getting vaccinated, you're going to get much closer to that herd immunity quicker than you are otherwise. And so I wouldn't be shocked if we start to see that, the real world data bears out, because like I said, I think Israel, has vaccinated a lot of their population, and they've seen some very nice results already within a very relatively short period of time with the Pfizer vaccine. I have no doubt that it's going to be reproducible with the other vaccines as well.
Yeah, absolutely. I just remember in early February, March, there was a lot of concern that we as humanity had never developed a coronavirus vaccine. And I just think it's an amazing testament to not ever bet against the power of technology and people absolutely committed towards a cause, right? Because and I think that second ingredient that you mentioned is really crucial. And we have so many other challenges that are obviously not as acute and worldwide, but some of them, many of the common and rare diseases that you've worked on are on a similar scale to this. And I hope that as a field, we can take some of that sense of urgency. Certainly don't need everybody working 80-hour weeks for the rest of their lives. But if we can learn what was it that allowed these to go so much more quickly and effectively and apply that, across the board, I think that it would be a real silver lining of this last 12 months.
Yeah, absolutely. I think the areas where we'll probably have some really good learnings around some of these things are with respect to how quickly the supply chain. I think that there'll probably be a lot of really good learnings on how quickly can you manufacture things and get them, have them be tested appropriately, have them be ready to, make sure that, they're fit for, obviously human consumption, all of those things. I think that there are going to be some huge learnings on that manufacturing side. And then as well on the actual conduct of the trials, I think that people have learned, how you can be really efficient with data collection. I'm assuming, I could be wrong, but I'm assuming that, in looking at the clinical trials and the data that they've, while they had a lot of data collection, they probably didn't have a lot of extraneous data collection. And I think that that's one of the things that bogs clinical trials down so much. You always want to know answers to so many different questions. You're collecting so much additional data that people are spending all of their time putting in all of this additional data that may or may not be germane to your question. And the challenge is, and I run into this problem myself all the time. If I'm designing a trial, I'm thinking, man, it would be really good to know what type of effect we have on people who might have, red hair, or,
right.
And then let's put in a hair questionnaire, right? And or whatever it is, you put in these questionnaires, you put in these things, and you're like, well, is that important to my question? Am I making it harder for the site to get the data in? Am I making it harder, is the patient having to be there for 2 hours as opposed to, being in and out in a half hour? All of these things that, if you don't think about it, it can actually really delay you're getting the data and the answer to the question you want. And so I always ask myself, is this important? Do we need it? is this really going to help us advance moving forward, or is it going to help the patients moving forward? And if it hits all of those criteria, then it probably is something we should do. I'm assuming for these trials, they were very, matter of fact about it, and said, look, we're going to have as efficient data collection as we can. We want to get patients. We want to get as much information as we can, the right kind of information, but we don't need to know more than that. And, and we can move on from there. And I'm assuming that, those types of decisions really help them with the speed and rapidity with which they were able to execute.
Yeah, I think that's a really good takeaway. And I was actually going to ask you what kind of framework you use to choose which questions are in or out, but I think you already more or less covered it, because the challenge really is those ones in the middle, right? There's some things where you say, if I absolutely need to know this at the end, and there are other things that are extraneous, but there's always stuff in the middle that if you do the exercise of a pre-mortem. I got in the habit of doing this. My PhD supervisor taught me to do this, and I found it to be really effective. Is that how you do it? And think about, maybe you could explain a little bit about how you go about actually designing a clinical trial and what your thought process is like.
Yeah. So I like the way you described that as a pre-mortem, because I do think that that's exactly the right way to think about it. It's okay. So, one, what are the key questions we want to answer? And are there some other additional questions that are important to answer? And then it actually helps you to, at least it helps me when I'm thinking about a trial. And I'm never the sole person who designs a trial. I always, and this is how I've been doing things actually even since my time when I was at Bristol Myers Squibb, is that when you're designing a trial, it actually makes sense to get the input of all of your stakeholders. So what I tend to do is try to get everyone in a room and just do some whiteboarding around, okay, we know we're conducting a trial. What are the key things we want to get out of this trial? Let's just write them down. Yeah, in broad strokes, let's write them down. What are other things we want to get? And I usually start that with some independent data collection, just so, because you never know. There are some people, like, I have commercial colleagues who may have some different thoughts as opposed to some of my scientific colleagues. And I just do it as kind of blinded data collection, and I just write everything up. There's no good answer. There's no bad answer. And then you just, and you talk about it as a team. Okay, how important is this? Is this something that's so important it should be a primary endpoint? Is this something, and you kind of go through that process, right? And it makes things actually very easy, because ultimately you get to a point where it's like, okay, I can answer, the team may want to have 20 questions answered, and everyone knows you can't answer 20 questions. So then you have to start making some decisions around those questions. And what I try to do with most of my, trials is I also want to have someone who's there to help represent the patient's perspective. Sometimes it's hard, because it's hard with rarer diseases sometimes, just because finding patient advocates who can help collaborate with you can be challenged. Just challenge from a timing perspective, finding people who have time to help you take time out of their busy day to work with you on this. But there are a lot of patients who are willing to give their time because it's so important to them, because they're so passionate about it. But you always want to have someone who can express that patient perspective. And if you're not able to get that, then I find it important to be able to go to the patient advocates after you've kind of had the framework for your study, so you can make sure that you're looking at the right types of endpoints, the right types of things that are important to them. But once you put that framework in, you realize you can't answer every question. So then you have to say, start picking and choosing. And we're very fortunate, right? We can answer a decent number of questions. So we can answer questions on biomarker endpoints. Usually, if it's a blood sample, we can answer questions with respect to actual clinical outcomes for some of these diseases, and we can talk a little bit about some of the diseases we're going to be doing at Aristea in a little bit. We can also answer questions about quality of life. If the patient, as long as it's not too onerous and the patients are willing to go through a quality of life questionnaire, I think that those are really important questions to answer, because that cuts to the heart of a lot of these diseases. Why we do it, right? So you can answer a lot of questions. You just have to be focused and say, okay, well, are there questions that aren't that I'm just not going to be able to answer with this clinical study. And sometimes that has to be okay. And you'll have to answer that with the next clinical study, or you'll have to answer that in phase 3 and maybe not in phase 2, or you'll have to just design a different study as maybe a smaller study to answer that question.
Yeah, 100%. That's really helpful. Maybe this would be a good segue to talk about what you're doing at Aristea and what kind of diseases you're working on. And also, I'd love to just talk about your personal transition from big pharma Bristol Myers Squibb, Roche into much smaller biotechs, which have probably different, very different differences in terms of what the day to day is like. The number of molecules or disease areas you're thinking about, but also the way you operate and the speed at which you can move. So I'd love to hear maybe about that general transition, and then a little bit about what you're currently working on.
That's a good area for us to talk about, because I had spent about 5 years at Bristol Myers Squibb before I decided to come to a smaller company here in San Diego. And I've worked at a couple of smaller companies here in San Diego. And I think the one thing that I really had while I was at Bristol Myers Squibb was you have all these resources, but everyone really focuses on their area, right? And that's something which is great. Within large companies, you really need specialists who can focus on things. I do tend to think of myself as a generalist, someone who likes working in different things. And I think that, when you're at such a large company, the focus always goes on the larger programs, the larger areas. And that was just something that, there was a component there that for me, just didn't fit with how I like to work. I really want to, have a direct impact and really push things along and forward. And within a larger organization, there is just a lot of bureaucracy. You need to, to get clinical trials designed and approved. There are committees and they're there for good reason. They're there to ensure that there's appropriate scientific rigor behind every, study that's designed. They're there to ensure that everyone agrees with how you're doing things, all the way up to even the chief medical officer at such a large company. They're there for more good reasons than bad reasons.
Right.
But with that being said, when you go to a smaller company and there's a focus only on maybe 1 or 2 assets as opposed to 20 assets, suddenly you're liberated to some extent.
Right.
And you can take some of those same really good practices of how you design a clinical trial, because really where I learned to design clinical trials was at Bristol Myers Squibb. And I took that experience and I applied it within the smaller biotech framework. And suddenly you're now quickly, because you don't have to go through 3 layers of management reviews. The management review stops at 1 layer, and maybe that 1 layer is actually in the room with you helping to design the study and giving you buy-in. But you're still using the same rigorous approach, right? You're still having the statisticians pressure test things. You're still, running things through the clinicians, the patient groups, et cetera. You're still doing all of those things that you learned, but you're doing it in a much faster manner. And as soon as within 2 months of my moving out here, my wife said something to me really funny. She goes, "Oh, you're never going back to a big company ever." And I said, "Why?" She's like, "Well, you work really hard, but you don't complain about work ever. You seem like you're enthusiastic about going into work all the time, even though you're there till whatever hours." And I said, "Yeah, I guess I hadn't even thought about it. I just, dove in and started doing the work." And, she just saw it immediately, within me, because when you're working with like-minded people and you're able to move things along and you can really pull together as a team, there's so much you can accomplish. And there's such a good feeling of being on that type of a team that you can really have an impact for patients. And you just want to keep recreating that feeling over and over again. And so that's kind of been my mantra, so to speak, as I've been going through the last 7 years here in San Diego, is just trying to recapture that experience and really trying to figure out a way to help patients.
Yeah, I love that. Maybe you could tell everyone a little bit about how a small biotech gets started, and maybe we can use Aristea as an example, because not everybody listening will have worked in large or small, and it'd be good to just talk about, even back to basics, what an asset is. How does one discover an asset and then build a company around it and decide which diseases to go after?
Yeah, so there are a couple of different ways you can find assets and start to develop them. We were very fortunate at Aristea. This was a molecule that that our team had actually had the ability to work with while we were at another company, Ardea Biosciences. That was the first company I moved to when I moved to San Diego. And Ardea Biosciences was a wholly owned subsidiary of AstraZeneca. So the molecule that we actually got we in-licensed it from AstraZeneca. AstraZeneca and large pharma does this all the time. Every company does this. They basically have, assets that they're bringing through the pipeline, and then, they have either, a decision, we don't want to develop in this area, or we don't want to work in this specific pathway anymore. And then suddenly they have these great molecules that are now sitting on a shelf for no really good reason, but they're just sitting there. And so this was a molecule we knew about. And so our CEO and founder, James Mackay, he actually worked with some venture capital firms, with specifically one, Novo Holdings, to help bring this asset out of AstraZeneca and spin out Aristea Therapeutics, which is what happened. So they got venture funding. So they got a Series A funding round from Novo Ventures, and AstraZeneca owns a small part of the company. That was basically the upfront payment was a small part of the company. And then we now were developing this molecule as we see fit. We have worldwide rights to the molecule. We have a few areas, that we're very interested in working in. And so we chose for our initial indication a very rare disease called palmoplantar pustulosis. It's actually a rare skin disease where patients get these horrible pustules that develop in flares on the palms of the hands and the soles of their feet. So as you can envision, it's quite challenging for these patients to do work, to walk. Just their quality of life can be quite poor. And unfortunately, there aren't a lot of really good therapeutic options for these patients, which is incredibly unfortunate. And we went back to the basic science of, well, what does our molecule do? Our molecule works by blocking the migration of neutrophils from the bone marrow out into the periphery, specifically to specific areas of inflammation. So these patients develop these pustules, and these pustules are neutrophil-filled pustules on the palms of the hands and the soles of the feet. They actually have a name as a class of diseases, and palmoplantar pustulosis is one of them. They have a name called neutrophilic dermatosis because it's basically a disease where you have skin inflammation that's full of neutrophils.
Full of neutrophils, right? Yeah,
exactly. So it's not it makes a lot of sense why this therapy might be very effective for this disease. And we have a host of other diseases that are also neutrophilic dermatoses, which are very challenging diseases, which we may develop in moving forward once we're through kind of our phase 2 for palmoplantar pustulosis. There's a couple of other diseases. One's pyoderma gangrenosum. Another one is Sweet syndrome. So there's a couple of these other diseases which are similar enough that we think that we would likely work in those diseases, but they're a little bit smaller in size, and we actually want to conduct as large of a trial as we can. And palmoplantar pustulosis has it's large for a rare disease. Right. We anticipate that there's probably about 160, 170,000 patients with the disease in the United States. So we can conduct a larger, well-run clinical trial and see, does our therapy work for this before we go into those smaller studies where we have to conduct a smaller trial. In those smaller diseases where we'd have to conduct a smaller trial, because again, you can get a false positive result, right? You have, we saw a great outcome, in this 20 patient study, when it may not really work. And so again, from the perspective of just thinking through this, for a patient's perspective, we really want to, do as large of a trial as we can do, answer the question appropriately, and then maybe we can go into some of these other diseases because we'll have that nice, really good foundation of evidence to move from.
Yeah, that completely makes sense. It feels like it's that deep understanding of the biology and how the molecule works that is really the unlock here. How did that come about? And has this been known for a very long time? And someone on, the founder or somebody else on the team managed to put all the pieces together, or is there a new piece of biological science that has kind of been built around this?
Yeah, so it's very interesting. This we know that modulating the pathway that we work on can potentially work in this disease. People have known about it for 10 or 12 years. There was an initial study that was done by an investigator in Europe who had used a monoclonal antibody to IL-8. So IL-8 is actually, it's a cytokine that's one of the CXCR2 ligands. And so, our molecule is a CXCR2 antagonist. So we block at the same area where an IL-8 antibody would kind of scoop up all the IL-8 and not let you potentiate anything related to IL-8. We kind of are on the flip side of that at the receptor side. We block any IL-8 binding, right, at the CXCR2. So they saw some positive results. Now, again, this is what happens in larger pharma. This asset was acquired twice by different pharma companies. Like one company, bought, it was like the fish eating the bigger fish, right? And this asset just kind of got lost,
right.
to the world, so to speak. And so, and we knew that this was out there. The state had been out there for about 10 years or so. And so the team thought that, well, that's a pretty good proof of mechanism here for us. Let's continue doing some development work along this pathway, but with a small molecule, not with a biologic agent. And I think that was one of the areas that we potentially saw that there was a lot of value for us, was a small molecule. And the difference between a biologic agent and a small molecule, just for some of the folks on your podcast, usually large molecules are injectables. You either get them by IV or you get them subcutaneously, and then they usually depot. They last for about a month or so, and for some patients that's great. Other patients are not happy seeing a needle in any way, shape, or form, and they like taking a pill. And so our molecule is a small molecule. It's a pill. It's an once daily oral pill that you take. And so we thought that this would be a nice mechanism for us to evaluate moving forward. So we picked PPP as our initial as our lead indication, but we did go into the science and really thought about some of the other data that we had. So it's very interesting. There's the team at Ardea Biosciences had conducted a few studies evaluating our molecule in combination with colchicine that suggested that the two molecules worked well together. And we looked at a few other diseases. And specifically, we looked at one disease called familial Mediterranean fever. It's a rare disease that I know because I took care of a few patients with this disease while I was at the Cleveland Clinic and at UCLA, not very many. UCLA, interestingly enough, as a center, has the largest population of FMF patients in the country, strangely enough, because a gastroenterologist there started seeing patients with this disease. So this is it's an interesting disease. It's a disease where patients will get really high fevers periodically, and they also have horrible abdominal pain usually, or chest pain. Like those are the hallmark areas, but they can also get inflammation of their large joints, their knees, their shoulders, their hips, etc. Their ankles. Like they can get a lot of swelling in those areas as well. And it's been under investigation for over 50 years. And the way that the people started using colchicine for it was because they looked at the fluid from the knees of these patients and the large joints of these patients. They pulled the fluid out and they looked at it and they said, this looks just like what a gout patient's fluid would look like. And we know that colchicine works really, again, going back to this was in the 60s, going back to how medicine was practiced then. Right. Well, if it works for gout, if colchicine works for gout, let's try it for this. Yeah. And it worked. These patients, it actually worked very effectively for these patients. So we were really interested in this disease because it's been under investigation for a long time. We know that colchicine works really well, but it only works really well for about 70% of the patients. And the challenge with colchicine is, and for anyone on your podcast who knows anyone with gout who's taken colchicine, they know a couple of things about it. Colchicine, one, can cause you to get really bad GI symptoms. Some people get nausea, some people get diarrhea. Like you can have some challenges with it. So getting up to an appropriate dose can be really hard for some patients with colchicine, and it's just challenging for them because it impacts their quality of life. For other patients, they can have other side effects from colchicine. They can have some problems with their white blood cell counts. They can have other challenges with their with taking a high enough dose of colchicine. And for some patients, they just it just, for whatever reason, they're intolerant to it. So there's a group of patients with FMF who, even though they're at what for them is a maximum dose of colchicine, they continue having these attacks. And these attacks can happen very often. So good control for a patient with FMF is having 0 to 1 attack per year. So complete control. You're under, and a lot of patients have very good control. But there's a proportion of patients, and with these smaller diseases, it's challenging to quantify the exact number, but we know that it's somewhere on the order of at least 20 to 30%, maybe as high as 40%, but unlikely that high, where patients have continue to have attacks. And we thought, well, if they're having attacks, that means they're not under good control. And the problem with FMF is if you're not under good control, you have this degree of subclinical inflammation that can lead you to have something called secondary amyloidosis. And you can get the deposition of this byproduct of inflammation into your kidneys and into your heart. That can subsequently cause heart failure or kidney failure, which is obviously a bad outcome over a long period of time. And people have shown very clearly that if you treat someone with at least 1 milligram of colchicine a day, you can reduce those subclinical levels of inflammation, keep the attacks away, and actually keep them from going on to kidney failure. So it's very clear that keeping people under good control really impacts their long-term outcomes. So we had seen a little bit of data that suggests that we know how colchicine works. Colchicine actually works by blocking the migration of some of those white blood cells from the vascular space, so from your blood out into the inflamed areas, whether it's in the abdomen or in the chest or in the joints. But for the people who aren't tolerating, who continue having attacks despite being on a maximum dose of colchicine, obviously the colchicine isn't working because they're continuing to have these attacks.
Right.
So we thought that blocking at a different site, blocking the migration of those neutrophils to those areas of inflammation, that in combination with colchicine might be actually able to ameliorate the condition for these patients. So that's our hypothesis. We have a little bit of animal data that suggests that that makes a ton of sense as well. And we know that this, there's a pretty high unmet medical need for these patients. Now, the challenge is it's a genetic disease. So the gene that's specifically impacted is a gene called pyrin. So pyrin normally functions to basically it's like a checkpoint, and it keeps your immune system regulated appropriately. And if it's not appropriately regulated, your immune system's constantly firing, and you get a hyper-inflammatory response. So these neutrophils just are going to wherever they want to go to.
Right. Our
thinking here is that if we can have colchicine on board in addition to our molecule, we can keep these patients under very good control. The subgroup of patients who aren't tolerating colchicine or are continuing to have attacks on colchicine. So that's our hypothesis. We're going to conduct a small study proof of concept study, but we're lucky in that a lot of other people have tried developing in this space, and we kind of know that you don't need a lot of patients to see if the drug works or not, because the patients can kind of act as their own control. I was having a lot of attacks before. I'm having zero attacks now.
We're doing something right.
Yeah. Something's happening, right? So that's we're going to conduct actually a placebo-controlled trial. So some of the patients are getting placebo for a period of time, but because they're uncontrolled, we recognize that we can't keep people on. It's not ethical to keep people on placebo forever,
right.
Or for an extended duration of time. So we're going to be running a slightly shorter randomized trial. So that the people who are on placebo, if they continue having attacks, they can subsequently be rescued and get therapy. But yeah, so we're really excited about that. This is a disease that's actually much more prevalent in certain parts of the world. So in the U.S., the prevalence is lower, but in certain parts of the world, like in Turkey, Israel, Armenia, some countries in North Africa, it's actually very common in patients of Jewish descent from North Africa, as well as there's a high proportion of patients in Israel. Israel actually has one of the best databases in the world of patients with FMF. They have almost 10,000 patients with the disease. So we're going to be conducting this trial likely in a few countries. Probably Israel, Turkey, maybe one or two other countries, and obviously in the United States as well. And we're really excited that we might be able to, bring a new therapy for these patients who are continuing to have problems.
What are some of the challenges that you've encountered in developing therapies for rare disease that are different than those in the more common diseases that you've worked in, or are there actually more challenges that are in common? I'd love to hear, a little bit of compare and contrast between the two.
Yeah, that's a great question, because I, in my last role, I was at another company where I worked in two very disparate diseases. I worked in asthma initially, and then I worked in another disease called pulmonary arterial hypertension. And even within that, it was incredible. Within asthma, it's almost like being in cardiovascular disease, right? I mean, there's just so many patients with it. You kind of have a template for how you need to run your clinical trials.
Right.
And you could try to collect some other data that will tell you things. You can do some pharmacogenetic analyses. You can do certain things to help you understand it. But the evolution of thinking and understanding of asthma has just, gone through the roof in the last 10 years. People have really understood now, wow, there's this thing called type 2 inflammation that's mediated by eosinophils. There's this entire, biology that people have brought to the forefront. And it's allowed people to develop new therapies that are amazing for patients with asthma. And that's the value of working in a large disease, is that you can actually conduct that level of science where you can actually start doing targeted types of therapies like that. Right. I think the challenges in smaller diseases is really in determining, in getting to that level of precision. I think that's really, the problem is not every patient who has any of these diseases we're talking about, whether it's pulmonary arterial hypertension, familial Mediterranean fever, not all of them have the same presentation. They don't all act the same, right? And so there's this spread in how they act, but there aren't enough patients for us to try to subsegment the population and understand what therapy is going to work for which patient. I think that for me is the biggest challenge, is that you can have a variable response, and it can actually potentially help you not answer the question appropriately, because if 60% of a very small population responds to your therapy, you're likely going to have, be able to see that. But maybe if only 20% respond, but you know who that 20% are, that could be very valuable for those patients. But are you going to get enough numbers to ever see that? Whereas within a larger disease, you can potentially find that. And I think that's the real big difference between, looking at diseases that are larger diseases as opposed to the rarer diseases. I think it's just a little bit more challenging.
Yeah, I think that's a really great point, because we're seeing I think it's fair to say most common diseases are being splintered into ever smaller and more targeted groups based on genetics, other biomarkers, understanding what's really going on. And we hear anecdotally, and there's occasionally research work on it from a rare disease perspective. And we certainly know every patient is different, but if you're struggling to get the numbers to fill a clinical trial in the first place because of the rarity, then it becomes that much harder to understand, actually, are there subgroups within this already ultra rare group?
Yeah, absolutely. And I think the challenge is for us is trying to answer as many questions as we can, because we know that there's so many patients who have a need, right? And when you work within this rare disease space, you're always trying to think about, well, how can I serve as many of those patients as I can? Whereas, in the larger diseases, you just have this momentum to serve all of those patients. And that's the one area where I know I'm always challenged with, is I want to try to serve all of those patients because I feel like all of them need something. And how do I do that? And how do, and then at some point you just have to step back and say, well, if I can help some of those patients, maybe I can help figure out the other, the group of patients that didn't respond to my therapy. Maybe we can figure something out for those patients. Maybe we can learn something about those patients, and then we can figure out a therapy that will help those patients. Or maybe there already is a good therapy that can help those patients that we can apply. So it is. Sometimes it can be disheartening if you get too caught up in that. But I always think about the fact that, well, we always want to develop where there's a high unmet medical need, because if there are enough patients who have a need, then you're doing good.
Yeah, no, I think that's a really great place to end, because we're running right up against time, and I've taken almost 54 of minutes of your precious day. So I want to say thank you. I've learned a ton just talking to you, and really appreciate you sharing all your experiences with the group. If people want to keep up with you and your work, I wonder if you could just share your website, if you use social media or anything like that.
Yeah, absolutely. So we're at aristeatx.com. That's our website. We will be posting some information shortly about our ongoing clinical trials. We'll have stuff posted on clinicaltrials.gov with respect to familial Mediterranean fever, palmoplantar pustulosis, and another disease we didn't get a chance to talk about, but which is also a relatively rare disease in the United States, Behçet's disease. we're really excited to be kicking off our clinical development in all of these diseases moving forward. We think that we really have a chance to help a lot of patients here. We have social media presence as well via LinkedIn. So please follow Aristea Therapeutics on LinkedIn. We'll have a lot of interesting things in the coming months around our clinical trials, as well as how we're reaching out to patients with palmoplantar pustulosis, familial Mediterranean fever, and Behçet's disease. And Patrick, I just want to say thank you so much for having me on your podcast. This was a lot of fun.
Amazing. My pleasure. Thank you so much.
All right. Thank you.
Thank you for listening to this episode of the Genetics Podcast. If you enjoyed the podcast, we'd really appreciate if you left us a review on your favorite podcast player, or even better, you can tell a friend who you think might like it too. As always, you can reach us anytime at podcast@sanogenetics.com. We really love to hear from you all about any feedback you have, guests you'd like to hear from, or topics that you'd like to see us cover in the future. Thank you again for listening, and we'll see you next time.